January is an exciting month, not only because it's the first month of the year, but also because Cathie Wood's Ark Invest issues their Big Ideas report. What is the Big Ideas report all about? It's about change and convergent. When has investing not been about the future? It reads on page 3 of the report. Change appears to happen slowly and then all at once. Over time, innovation should displace industry incumbents, increase efficiencies, and gain majority market share. With the right understanding of disruptive innovation and a long-term horizon, we believe investors will capture exponential growth opportunities which deserve a strategic allocation in their portfolios. For this reason, Ark focuses on opportunities likely to scale as technologies converge, transforming entire industries. To enlighten investors on the impact of breakthrough technologies, we began publishing Big Ideas in 2017. So it's the sixth installment. This annual research report seeks to highlight our most provocative research conclusions for the coming year. We hope you enjoy our Big Ideas for 2022. Great words at the beginning of the report. And the great thing is, of course, they also cover the blockbuster topics like battery, robotics, EVs, blockchain technology, but also the life science area. And the amazing thing with this report is that life science is explained in an easy to understand way for non-scientific investors. When I go in the report on the page 84, we see the first area, Multi-Omics, the future of molecular biology. And in this video, you will hear Simon Barnett, Ark analyst, who leads you through the coming pages. What is multi-omics all about? Leveraging the genome, transcriptome, proteome, and other omics, scientists are likely to translate groundbreaking discoveries into clinical solutions, which means at the end of the day, products for patients. New analytics and software tools, especially those focused on high-throughput proteomics, should allow scientists to unlock the codes to life, disease, and health. According to Ark's research, the multi-omics revenue opportunity, including life science tools, basic and translational research, population health efforts, and molecular diagnostics, will scale at 22% annual rate from roughly 110 billion to 300 billion during the next 5 years. Multi-omics is the first part, and the second part in this report is one of my favorite parts. As one of the companies in its gene editing, has also a strong connection to my home country, Austria. It's CRISPR Therapeutics. And gene editing with the nice slogan, curing disease, not masking symptoms. It's a great approach. In this video, you will see the second analyst. It's Alexandra Urman, who will lead you through this part of the presentation. And gene editing is the convergence of next generation DNA sequencing. CRISPR gene editing and artificial intelligence has the potential to transform healthcare. These advances could accelerate the pace of scientific discovery, personalizing medicine to cure disease instead of masking symptoms. Based on Ark's research, the equity market capitalization of gene editing and gene therapy companies could grow 54% at a compound annual rate of return, scaling from roughly 130 billion today to an amazing 1.1 trillion by 2026. But not enough when we look further in the future until the year 2030. Ark Innovation expects that the area of gene sequencing will grow from 125 billion in 2020 to 3.6 trillion in 2030. Huge potential. And I recommend listen to the two analysts from the Ark research team and enjoy the video. Have a great evening.
Hi, everyone. Thanks for joining. My name is Simon. I'm an analyst on Ark's Genomic Revolution theme, where I focus mostly on life science tools and diagnostics. I'm really excited to finally be able to share some of our research on multi-omics and the future of molecular biology. So let's get started. In previous years, we have focused on genomics, the study of DNA. Today, I'd like to take a step back to discuss the study of RNA, called transcriptomics, as well as proteins, which is called proteomics, which I'm going to focus on pretty heavily. We believe that the future of molecular biology is going to be powered by these multiple omics categories, how they interact with one another, how they influence each other to affect life, disease, and health. So we think that the revenue opportunity for multi-omics is vast, encompassing everything from, again, life science tools and hardware to the associated software, applications in research, whether it's in government, academia, or translational research into the clinic. Altogether, we think that these technologies and the applications thereof should scale at roughly 22% annually, from about $110 billion this year to over $300 billion over the next half decade. So let's start off at square one with DNA, the Human Genome Project. Contrary to popular belief, the draft sequence that was published in 2003 was not complete. The Telomere to Telomere Consortium, which was an international cohort of scientists and researchers, leveraged long-read sequencing from both PacBio and Nanopore to finally complete the genome, discovering hundreds of millions of base pairs of DNA and roughly 1,500 new, quote unquote, genes that we're just beginning to make sense of today. Now, the point of this isn't to disparage short-read sequencing, which was the technology that gave us the original draft sequence, but rather to suggest that the same hammer can't be used on every single nail within biology. New technologies and methods, such as long-read sequencing, are surfacing new biology at a breakneck pace. So here I've listed 3 areas of discovery that we are seeing empowered by long-read sequencing. Namely, those are the detection of structural variants, which are a significant class of variation within humans and other organisms that long-read sequencing detects much more aptly. Epigenetics or methylation, these are small chemical modifications to the exterior of DNA that doesn't alter sequence, but changes the way that your body interprets its own instructions. And finally, something called long-range phasing. I'm sure many people know that we inherit some DNA from mom and some DNA from dad. Chromosomes come in pairs. And so the ability to understand whether or not genetic variants are on the same side or on alternate sides, right? So on mom's or dad's side. These are areas of biology that we're just beginning to understand. And it just really goes to show you that even the genome still has new ground that we're breaking. So as alluded to, let's expand our horizons beyond just the genome to talk about the transcriptome, which is RNA, and the proteome, which is proteins. Together, these 3 bodies of code are often referred to in something called the central dogma of biology, which crudely describes the directional flow of information from DNA to RNA to proteins, and finally to one's phenotype, such as disease state or health or predisposition to certain conditions. And I've illustrated that here with a cartoon that actually shows a single human cell. You can see how the information moves the same way that you read, from left to right, and changes from DNA to RNA to proteins. And if there's one thing that you should take away from this slide, it's that these 3 categories of molecules are interlinked. And the more that we discover how, the better our ability to prevent, diagnose, and treat various diseases. I want to focus quickly on proteomics. Specifically, I'd like to focus on scalable clinical proteomics, meaning the application of the technology for the betterment of diagnosing and treating diseases. And I want to focus on this for a couple reasons. Firstly, the proteome is a really tough problem to solve, and throughout this presentation, you'll see reasons as to why. Second, solving the proteomics problem carries an enormous biological and commercial potential. And we believe that there are several disruptive technologies that are boiling to the surface today that may allow us to really make a dent in one of the most difficult problems in biology. So first of all, clinical proteomics is really hard for for one reason being that it's limited to blood plasma in terms of one of the only real feasible unbiased sample types in humans. So let me explain that a little bit more. Sampling blood is a great way of looking at the proteome because it is a global reporter of what's happening inside the body. What I mean by that is if you take a liver sample versus a lung sample versus a brain sample, the proteins are going to look very different. However, virtually all organs and organ systems will leak proteins into the blood. That's why taking a blood sample is a good, like I said, unbiased reporter. And I'll explain some of the challenges with this because it's not quite so straightforward. If you look at the chart in the bottom left-hand side, you can see that proteomics technology hasn't declined in cost the same way that DNA and RNA sequencing have. In fact, you can see the red line, which represents proteomics, really only falls in a staircase style a couple times over the past 20 years or so, each of these corresponding to a new piece of hardware technology that gives us a view and a method for detecting proteins. But as you can see on the right, each of these colored dots represents a new technology that we think could enable a similar precipitous cost decline, like a little bit like what happened to NGS or next-gen sequencing for DNA and RNA back in 2008. So you can see here on the right several new technologies such as those created by Olink, SomaLogic, and Nautilus, we think are going to really accelerate the cost decline and increase the accessibility of the proteome. So I want to give a special mention to the workhorse of proteomics today. This is technology called mass spectrometry. It's ubiquitous. It's everywhere. There are over 10,000 units out in the field today across the world, but the technology is quite hard to scale. However, we do think that you can teach an old dog new tricks. So similar to DNA and RNA sequencing, mass spectrometry isn't necessarily plug and play. It's a process. It involves methods that live both upstream and downstream. And we like to refer to those internally as bookends. And you can see them here represented on the left and right-hand side. So over the past few years, we've seen a tremendous amount of what we call bookend innovation. That is, novel sample prep techniques or software packages that live upstream and downstream of mass spectrometry and that are really allowing it to be revitalized, which is terrific, like I said, because since these things are already in the field, we think that they can be, adopted in much more short order, than otherwise would be the case. So on the left, you can see a chart of the concentration of proteins in human blood plasma. This is one of the biggest issues with using plasma to study the human proteome. And what I mean by this is that the most abundant proteins in blood and the rarest proteins in blood are separated by over 10 orders of magnitude in concentration, making for really difficult signal to noise problems, right? So, being able to see those needles in a haystack. And I'll point out here, just because a protein is rare doesn't mean that it isn't important. Nanoparticle technology from companies like Seer are enabling both common and uncommon proteins to be detected by mass spec at the same time, just with a sample prep innovation happening on the front end. Meanwhile, other technologies like swath, and you can see that here in the middle, take advantage of certain types of metadata that comes off of mass spectrometry instruments, which enables them to be both faster and more accurate. Finally, open source computational packages like OpenPip, which you can see a chart of on the right, this is a technology powered by deep learning, really helps to accelerate the oftentimes tedious and manual analysis of mass spec data, which frankly is still a little bit artisanal and manual. And you can see how well the neural net performed to PhD level human annotation there on the right. And I'll move on to talk a little bit about some of the newer detectors, the newer technologies, the ones that are not mass spectrometry, but are really enabling a much deeper, wider view into the proteome than ever before. And this is a really amazing chart. It dates back to the early 1930s and is representative of state-of-the-art technology at each time point. So the y-axis, which is in log scale, shows the percent of the proteome that is undetectable by the leading technique at that time. So the closer to the top, the better. As you can see, the improvement really moves along at a snail's pace up until the mid-2000s, until several new methods, and I've called out those here, SomaLogic and Nautilus, potentially enabling us to see, again, potentially north of 50%. And we think by the end of 2024, scientists will be able to see the entirety of the human proteome the same way, you can see the entirety of the human genome today. We think this will be within reach of scientists within the next few years. And we think these technologies are much more scalable than other approaches. Quickly moving on, I want to talk a little bit about the complexity in the central dogma. So we're taking a step back again and considering DNA, RNA, and proteins. And if you take a look at this chart for a moment, I think that there's, I want to emphasize that there's a difference between detecting a molecule and fully understanding it in the context of what's happening inside of the body. So I'll call your attention to that solid purple band, the very thin one at the bottom that runs left to right. You can see that the 20,000 genes that make up our genomes do indeed move downstream to create what are called 20,000 canonical proteins. However, there's a much richer truth behind this. That is that there are things like regulatory processes, mutations, chemical modifications, all of these things happening in tandem to create much more complexity as we move left to right. And you can see this chart's actually drawn to scale. The lighter purple section that's moving up and to the right like a ramp shows you that, if you look at the right-hand side, even though there are only 20,000 proteins, there are over 1 million variations of those proteins, which we think only nanopore and semiconductor-based sequencing technologies are really capable of deciphering. And we think that the maturity of both semiconductor and nanopore technologies are at a point where we can start to make these discoveries and really meaningfully translate them into clinical practice. I'll be very quick here on this slide. It's the same that I've shown earlier about the central dogma. And I want to just kind of put this in here to illustrate the fact that sometimes a new perspective on an old problem is all that you really need. And what I mean by this is that the more that we master each of these domains of biology, DNA, RNA, and proteins, we can almost back solve or look at a problem from a new perspective. So in this yellow section, I've talked a little bit about some of the population scale projects that are going on, where we're marrying together data sets that contain genomic and proteomic information to help really expose a lot of the underlying mechanisms that previously were very difficult to look at. And then finally, with long read sequencing directly of RNA, not only can we sequence entire RNA molecules and directly compute the protein sequence, but we can also look back to understand how a really key regulatory process called alternative splicing gives rise to biomolecular diversity and potentially how it's involved in the formation of various diseases. So to end, I want to talk a little bit about the market opportunity here. I think, again, this is a situation where we're really considering the combination of the genome, the transcriptome, the proteome, the associated life science tools, and the applications of these tools across many different end markets, whether it's for research, for translating something into clinical practice, or applying it in the clinic globally. So altogether, once again, we think that these technologies will compound roughly at 22% annually from $110 billion to roughly $300 billion in revenue opportunity over the next 5 years. And the last maybe point to mention is we think the largest categories that comprise this forecast have to do with, first and foremost, oncology, detection, therapy selection, surveillance, monitoring, screening, everything in between, the non-invasive monitoring of organ health, whether it's cardiovascular, neurologic, transplants, things like that, as well as some population health efforts that are really picking up steam today. And this is actually one of the main reasons why I wanted to focus on plasma as a sample type, because liquid biopsies, that is, using sequencing or proteomics technologies to scour the blood for signals of disease, it's really important that all of these technologies be compatible with blood samples. And so that's, again, one of the reasons why that was, an emphasis in the presentation. So thanks again for your attention, and stay tuned as we publish a little bit more on this subject. I'd be happy to discuss with anyone further on Twitter. You can follow me at @SBarnettArk. Thanks again for your time.
Hey, I'm Allie Urman. I'm a genomics analyst at Ark Invest. I cover therapeutics, including gene editing, gene therapy, immunotherapy, stem cells. And today we're going to talk about gene editing, which is one of our big ideas for this year. So let's do a little deep dive here. So we've spoken about an importance convergence, and that is the convergence of DNA sequencing, artificial intelligence, and CRISPR. Let's dig a little deeper on artificial intelligence. What does that include? So that includes machine learning, deep learning. It also includes things like AlphaFold, which is a neural network-based algorithm that can predict protein folding. And this whole convergence can actually revolutionize healthcare. And so we've also discussed this a little bit previously, and that is that this convergence can accelerate market improvement by 25%, and it can decrease failure rates by 25%. Both of these then will accelerate delivery and timelines, and it can create a plethora of new therapeutics that could be sent to the FDA for potential approval. This really opens up a new class of therapies, gene editing therapies, gene therapies, and they're really starting to get into the clinic now and looking to get approval. An example of this is indications like hemoglobinopathies, like sickle cell disease, beta thalassemia, and even TTR, which we saw really great data for, first in human in vivo data in late last July. Because of all this, and based on Ark's research, the market caps of gene editing and gene therapy companies could grow 54% at a compound annual growth rate, and that could scale from 130 billion, what it is today, to over 1 trillion in 2026. Now, let's take a step back and try to understand all of the factors that are contributing to the innovation happening here in gene editing and gene therapy. So first, we'll talk about the pace of gene editing and innovation and how it is accelerating. So there are 3 main gene editing nucleases that we talked about. So zinc finger nucleases, TALENs, and CRISPR. Zinc finger nucleases use engineered zinc finger repeat domains to target specific sites in host DNA, and they induce double-stranded DNA breaks with the nucleus. If you look at the chart here, it took about 8 years from its first eukaryotic edit to the time of its first human ex vivo application. So let's unpack some of the terms there. What is a eukaryotic edit? Well, it's an edit of any organism or cell that has a clearly defined nucleus. However, since we're talking about not human, another way to say this is that this would be the first successful gene edit on a non-human eukaryotic cell using the specific gene editing method that we're talking about, which in this case is zinc finger nucleases. Let's now unpack ex vivo. Ex vivo is when the editing is taking place outside of the body, whereas in vivo is when the editing is taking place inside of the body. For more information on the differences between in vivo and ex vivo, you can look at our big ideas from last year where we talked about the shift from ex vivo to in vivo gene editing. As new nucleases were developed, TALENs, CRISPR, and even new functionality was added to the CRISPR nuclease, you can see that innovation timelines really accelerated. So in the example we just gave, we said it took 8 years for zinc fingers to do what CRISPR could essentially do in 3 years. We believe that these timelines could get even shorter in the future as new developments and new improvements continue to proliferate. We're going to have a discussion with David Liu, and we'll talk about base and prime editing, which were both developed in his lab, and how quickly Andrew Anselon, who's a postdoc in his lab, went from conception to first successful experiment. And the acceleration there was pretty remarkable. In addition to speed, though, these newer nucleases, or ones with increased functionality, may be also able to address more clinical variants, which means that they'll have the ability to address even more diseases with potential cures. Currently, we believe base and prime editing could address many more variants of concern. And as you can see here, prime and base editing, which are the CRISPR derivatives, can address even more diseases with 79% and 59% respectively. So as mentioned, CRISPR may be a superior gene editing method for many diseases, indications, and experiments. However, it's not a one thing fix all, so not for everything. But we described what a zinc finger nuclease is. Maybe now let's define what a TALEN is. So a TALEN, or transcription activator-like effector nuclease, what it does is it targets the host DNA using a unique modular protein, and similar to zinc finger nucleases, it induces a double-strand break with the nuclease. CRISPR, on the other hand, stands for clustered, regularly interspaced short palindromic repeats, and it uses a protein RNA complex to guide its nucleus to a target site, and if used alone, also causes double-strand DNA breaks. We'll outline a little later why that could be problematic. Here, however, we outline why CRISPR may be a superior modality for some things, although not for everything. We will get into that a little later as well. One, it's cost-effective. It's quicker to make a construct, so it takes days instead of weeks or months like with zinc finger nucleases. It's pretty easy to use, so there's not much technical difficulty. It's an RNA-DNA interaction, which is less complicated than a protein-to-DNA interaction. Delivery is also really important here. And delivery is easier because it's not two proteins around the target. Instead, it uses a guide RNA to get the nuclease to the specific site in the genome where it needs to travel to. Lastly, let's touch on multiplex editing. So multiplex editing is doing more than one gene edit, and it's much more difficult with zinc fingers or TALENs. And that's because CRISPR is able to engineer. It's easier to engineer. It's easier to get it where it needs to go. As you can see, CRISPR here is shown in purple, TALENs are shown in green, and zinc finger nucleases are shown in red. What we're showing here is sort of the proliferation in CRISPR, both in academic journals, in papers, in publications, and also in clinical trials. If you look at the chart on the left, you can see that in 2012, which is when the Emmanuel Charpentier and Jennifer Doudna paper came out, the proliferation of the papers just continued to accelerate after that. They also later won the Nobel Prize for this discovery, and it's continued to proliferate since then. In terms of trials, this started around 2015, where we see a strong proliferation of CRISPR trials based on its ease of use. Although CRISPR's functionality does extend beyond just gene editing. So as you can see here, it can do transcriptional activation, meaning it can turn on genes that are dormant or not expressed. It can do transcriptional regression, which means it can silence genes, for example, if something may be toxic. It can also turn genes off or on, known as epigenetic control. And what's interesting is also diagnostic imaging. So diagnostic imaging could be helpful. We've seen it be helpful during the COVID-19 pandemic because it's cheap, it's easy to use, you don't need as much infrastructure. So it can be very helpful, especially with the thought of third world countries. But back to the heart of what we think are really important use cases of CRISPR, which of course is gene editing. So by increasing functionality for CRISPR, we may be able to treat conditions that we previously thought couldn't be treated. CRISPR, all the way to the left, in the picture you can see the green there is the nuclease, the black on top of it is the guide RNA, and you can see that the guide RNA will take the CRISPR to the exact location that it needs to go to. Here we just talk about some of the differences. So CRISPR is relatively small, which is a positive. It's about 4 kilobases, and it has a specific PAM, or a protospacer adjacent motif dependence. What that means is that it needs to have a specific landing spot in order to get where it needs to be in the genome. If there is no PAM sequence near the cut, then engineering may need to be done, if it's possible, to get the nuclease there. This PAM sequence is typically around 3 to 4 nucleotides downstream from the cut site, which could mean that you may have trouble getting a nuclease to a particular region of interest. And as mentioned previously, zinc finger nucleases, talons, and CRISPR create these double-stranded breaks or cuts, which can cause indels. It can also cause a lot of other byproducts, which could potentially cause unintended consequences and potential off-target edits. Using CRISPR alone will get fewer addressable clinical variants because obviously CRISPR alone is great for disrupting genes. So an example of that is like sickle cell, where you want to disrupt adult hemoglobin to turn on fetal hemoglobin. But that may not be the right choice for addressing single base pair changes, insertions, or many others. Base editing is a way to correct misspellings or base letter changes within the genome. The size is a little larger, and it requires specific landing spot within the genome or a PAM. But this creates single-stranded nicks as opposed to double-stranded DNA breaks, which should help with off-target edits and, of course, can address a larger amount of disease variants. If you look at the picture here for base editing, we have the same green, which is the nuclease. We have the RNA guide, which will take it to where it needs to go. And in purple, we have a deaminase. The deaminase is going to cause the chemical conversion of one base to another. Lastly, for prime editing, which is the newest innovation, it's slightly larger, but it has a relatively low dependence on a PAM, which is the specific landing spot for the genome, which means that it can get to more sites and it expands its ability to treat even more clinical variants or mutations. This makes sense because if you're able to get to more areas of the genome, it, of course, also only nicks one strand of the DNA, like base editing, which should help with off-target editing and increase functionality as we don't need to just disrupt the genes. When you look at the picture here, we again have in green the nuclease. Then we also have an RNA, which in this case we call a PEG RNA or a prime editing guide RNA. And in purple, we have the reverse transcriptase as opposed to a deaminase, which we use for base editing. And a reverse transcriptase is an enzyme that can generate DNA from an RNA template. So just to close, as we mentioned at the start of the presentation, gene editing and therapy companies could reach over $1 trillion in market cap by 2026. The chart here shows in green the gene editing and therapy market cap as a percent of the pharma biotech enterprise value, and in purple denotes the same for gene editing and gene therapy, but for R&D. Gene therapy and gene editing could offer real cures for disease, rather than masking symptoms or perhaps giving a little bit of relief, but not a cure, as many therapies today offer. Because of this, we could see a rise in their funding, in clinical trials, in research, and even in approvals. So R&D spending of these types of therapies could actually rise from 3 to 17% by 2026 because of the promise of these innovations, but also because of the fast pace of innovation. In addition to R&D expenditures, as we mentioned, the market caps could command a 54% compound annual growth rate and could be from roughly 130 billion to what it is today, for it can get to 1 trillion in 2026. Thanks very much to everyone for listening, and stay tuned.